it's really a delight to open the conference in what is an incredibly exciting area of biology. And what I wanted to tell you about today was really some of the science that underpins the successes, the remarkable successes behind the immunotherapy advances that are being made right now. So what I'm going to do is I'm going to give you a slightly condensed version, since we're pushing the program back a bit now with the technical issues, but I will make sure that you get the take-home messages. And I think what is very important for you to see what these cells look like when they're doing their killing. So here is one of the cancer assassins. It's a cytotoxic T lymphocyte, and it is able to kill cancer cells with remarkable specificity and with remarkable accuracy, giving just the right amount of killing to destroy the challenge that it's met. So how does it do that? That's what I want to tell you about. So it really is a wonderful field to be working in at the moment because of the tremendous effects that the new immunotherapies have had against cancer. It is a cancer breakthrough, and there are so many opportunities now for immunotherapies that it's exciting to be working in this area. So just to remind you of the two different types of immunotherapies that are being used, there's CAR T cells, where in essence what happens is that you take the patient's own T cells and you put in a receptor which artificially is able, or has a receptor that's able to recognize a cancer antigen. And you give it a sort of tail that would allow it to signal and tell the T cell to kill when the receptor binds. And that's made up of a sort of medley of bits of the normal T cell receptor that normally will recognize antigens. The other way is to use checkpoint blockades, which are antibodies that basically stop negative signals, stopping the T cell receptor from signaling, if that makes sense. I think it's less well understood exactly how all of that works. There are a lot of labs working on it. But what is clear is if you block the negative signals that are stopping the T cell from killing using antibodies such as anti-CTLA4 and anti-PD-1, then you can get very good killing going and remarkable effects in cancer treatments. So what I wanted to focus on today was how do you make the best killer T cell? And there are two areas I want to talk about. One is, how does the strength of the signal the T cell gets, how does that affect its ability to make cytotoxic T cells? And how do those, what are the molecular mechanisms that control the killing once those cytotoxic T cells have been generated? So to take you back to the beginning, there are 5 million potential killer cells in a teaspoon of blood. You can see they're highly motile cells, and they're able to patrol the body and identify cancer cells that need killing. So how do you harness them so that they can do their job to the best of their abilities? So what happens when a cytotoxic T cell recognizes a cancerous cell is it makes a very tight interface with a cancer cell, which is also known as the immunological synapse, and then it releases the contents of these rather unusual lysosomes in the cell, known as secretory lysosomes or secretory granules, and they contain some very special proteins known as perforin and granzymes that can rapidly destroy the cancer cell. The way in which that happens is, here's a cartoon version, because you couldn't see it otherwise, that here are your secretory lysosomes. Once they come to the contact site between the T cell and the cancer cell, is they release their contents, and the perforin is able to bind to the membrane of the target cell and insert across that membrane and make a donut-shaped pore through which granzymes pass and trigger apoptosis. These secretory granules are delivered very rapidly to the point of contact between the T cell and the target cell, because they move along little microtubules that are shown here in green, and you can see that there's a focal point where the microtubule organizing center is all clustered together and all the microtubules converge. And so by moving down these microtubules, these secretory granules are all delivered to the same place right at the point of contact with the cancer cell. So if you look at that in more detail, what you find is actually that the microtubule organizing center consists of the two centrioles and is in a structure surrounded by many different proteins that give this electron-dense cloud, and that's a structure known as the centrosome. So we asked, is it just when the centrosome gets to the point of contact? Is that when all the granules are delivered? And is that really the key to finding the right spot where secretion needs to take place? So we used a marker that just recognizes the centrosome, and that's shown here in red in this green cytotoxic T cell. And this is the tumor cell in red. And if you follow this little red dot as the T cell meets the target cell, then you will see that actually, here's the red dot, it moves up to the point where the two membranes actually are in contact, and almost immediately the target cell is killed and starts blebbing. So this is because the granules have been delivered to the point identified by the centrosome. So it's a rather unusual mechanism of secretion, but it gives very, very precise killing. So to show you that in it, sort of all pulled together into one movie, Alex Ritter in my lab captured this very nice movie where everything happened in a single cell. So here is a cytotoxic T cell in green, which is the actin, and in red are the secretory granules. The target cell is shown, the cancerous cell is shown in blue. And then the centrosome is in blue, which is a bit hard to see, so we put a little sort of golf ball there. So you can see that as the cytotoxic T cell meets the target, that there's a very tight interface formed between the two cells with actin covering that interface. But then the actin depletes, and at that point, you can see the centrosome coming forward with all the granules clustered around as they move down the microtubules, and it gives this exquisitely precise delivery of the lethal hit, sealed by the actin around it. So it explains why the killing is so precise and so accurate. So how do you then think about how to make the best CAR T cell? So you can develop a ligand binding domain which will recognize the surface antigen, but should you make it a strong recognition or a weak recognition? And then what should you put in the tail to give how strong of a signal inside of the T cell to get the best outcome? So one of the things that you can do is in immunology is you can ask these kind of questions, because there are really wonderful systems that have been developed over the years by many different labs, where, for example, they have made a mouse that has a single T cell receptor, and that it allows you just to bind the little peptide that can be presented to that T cell receptor, and it can change the strength of the interaction between the T cell receptor and the peptide MHC quite dramatically and with very functional outcomes. So here is a peptide that binds to a T cell, binds to an MHC that's recognized by a particular T cell receptor in this mouse. And if you change just the fourth amino acid, then you can have either a very strong signal, or if you change glycine, it's a very weak signal, or you can use an irrelevant peptide that shouldn't give any signal at all. And then you can begin to ask questions like, okay, how does the strength of signal change the ability of the T cell to recognize the target, and what functional outcome does that have? So the first thing you need to know is that actually T cells are not born killers, and that the naive CD8 T cells circulating in your blood are mostly not activated. But when they see a pathogen or a cancerous cell, then they recognize something that's foreign, then they become activated, and over a matter of days, they develop into these pre-armed killer cells, which have the cytoskeleton and the granules, which then are able to undergo a very rapid killing that you've just seen in the movies in a matter of minutes. So this takes a matter of days, this takes a matter of minutes. So the two questions are, how does strength of signal change the generation of killers, and how does it control the rapid killing? So in order to look at how it affects the generation of killers, we looked at the transcriptional signature generated by different strengths of killing. And I'm going to minimize this slide because what I want you to take home is the punchline. But in essence, we looked at what the transcriptome signature was for cells which was stimulated either with a strong signal or a weak signal. And this was the work of Ariane Richard in the lab. And so you can see that if you then cluster the similar transcriptional output, the cells that give that, then you can see that all of the ones with the strong signal cluster together, meaning that they're expressing very, very similar RNAs. Whereas those with a weak signal sort of are more distributed across this trajectory. The important message that emerged from this study was that regardless of the strength of signal, we could see that even the weaker ones went along the same pathway as the medium ones in green and the strongly simulated cells in red. And that they all, in fact, if you looked at those that became fully cytolytic, which are those in the circle here, you could see actually all different colors were there. And so this means that actually they, regardless of the signal strength, they all give rise to activated cells and they all become fully cytolytic. So this told us something new. It told us that actually the signal strength isn't determining different pathways for them to take. It tells you that they're all taking the same pathway, but it controls the rate at which they take that pathway, the rate at which they're initiated. It's as though you've got a race, and so you release them at different times when you've given a weaker signal, whereas the strong signal, they all rush off at the same time and do the same thing. So that's interesting immunologically because it tells you that you can increase the range very dramatically by using the rate at which you activate to extend that range. But from your point of view, from the point of view of immunotherapies, what it suggests actually is that the strongest signal is not necessarily the best. Because if you give a strong signal, all of your cells are going to do the same thing instantly, and that's it. It's going to be all over. Whereas if you give a weaker signal, then there's going to be a slower release as they activate at different stages, and you might have a more continued killing. And I think that's something that is emerging from the clinical data now. So you can look at the same thing, not just looking at the RNA transcripts, but you can look actually at the signaling that takes place inside of the cell. And you can do this by a method called CyTOF, which allows you to label many different activated proteins within the cell using different heavy metal isotopes. But without going into the detail, what that allowed Claire Mara, a clinician PhD student in my lab, to do was to look at various different steps of the downstream signaling from when a T-cell receptor is activated and say, how does that change when you change the strength of the signal? So this looks very complicated, but it looks even more complicated here when she then shows all of the level of variation and all of the different events that are occurring in each of these cells. Each column is a particular phenotype. But what you can do when you do this kind of analysis is you can identify 4 different clusters of cells which seem to vary according to whether you vary the strength of the signal or the length of the signal. And then you can use 4 markers that are typical of that to study a population of cells. Say, how do these populations vary? Because they're clearly varying when you vary the strength or length of the signal. What's changing? So using these markers that Claire and Ariane identified, we then looked at how these cell populations varied. And what you can see if you just focus on these curves here is you can see that with the strong, the medium, or the weak signal, that actually for each of these different phenotypes defined by the different markers here, that they're all actually doing the same thing. They're going up at this time and down at this time, but they're doing it, it's a different proportion of cells. There are far fewer cells doing that with a weak signal compared with a strong signal. So again, what it told us is that actually all stimuli activate the same phenotypes, but they do it with different rates. So again, what they've done is they're changing the rate at which they start signaling, the rate at which they start transcribing genes. And that's what's then allowing them to control the difference, or to interpret the different strengths of signal. So what this tells us is that regardless of the limited repertoire of receptors that these cells will all have, and they'll have a finite amount of signaling machinery inside, they can actually make a tremendous breadth of responses because they can respond to different signals by varying the rate at which they activate. It's like bringing time into the equation to expand your range. And the same thing's happening. I think it's very beautiful. It's happening both at the transcriptional level and at the signaling pathway activation level. But the take-home message is the same. It suggests that actually, because they all get there, so even the weakest signal will produce a fully cytolytic cell, and it will signal the same way, but it will just take longer. And so it means that the strongest signal is not necessarily the best when it comes to immunotherapy. So that tells us how signal strength varies the generation of killers. But what about how you control, once you've got a killer, how does that control the rapid killing that's then seen? So again, we use the same mouse model to look at this, and we could see that you get, if you look at the number of cancer targets that are killed with a strong signal in red, you get lots of killing. With a weak signal in blue, you get much, much less. So what does that look like when we look at the cells? And so that's shown here in this video, where in the top video, we're showing the strongest strength of signal, and this is a T cell where we've labeled the granules with little red balls to show that where the granules are, and the blue shows the centrosome, and in white is the cancer cell in this case. And you can see that the T cell comes in and it recognizes the cancer cell very rapidly, and it focuses everything, delivering the granules very precisely and very rapidly to the target, and that will result in a very effective killing. What happens with a weak signal is that actually the recognition is not very good, and so the cell sort of touches on several different targets, and even when it eventually settles on one target, you can see that the polarization of the granules inside is a bit chaotic and not very effective. So there are many different levels which Gordon Fraser in the lab had studied to look at each of these different steps and how each of them was varied with signal strength. But I'm just going to focus on one of them for the purposes of the talk today. And that is, how does a T cell control its killing? So here you've got your cytotoxic T cell, and you can see its actin cytoskeleton that forms a cortex around the cell, and it's grabbing onto the cell using actin protrusions. But how does it control how many granules to release in response to different strengths of signal? So it turns out that actually it's the actin that is doing that. And so here's a T cell that is moving along a substrate, and you can see it both side on and looking down on it. And you can see these little actin projections at the front as it moves forward. This is typical of how motile cells move. And so I'll show you one more time. So if you think about it, it's those little actin projections that is going to be where the T cell will meet its cancer target. And but what happens then to the actin when it does meet the cancer target? So this shows an encounter between cytotoxic T cell and cancer, the cancer target in blue. On the left, you can see that there is the whole video. And in the middle is just a very thin slice through the actin, so you can see very precisely what happens to it. And on the right is the en face view, where it's as though you are the cancer cell and you're looking at the T cell coming towards you. And what you'll see is that actually the ruffling of the actin clears towards the edges, and so you can see here that you've got the actin is focused here around the edges, but it seems to be depleted in the middle. And in fact, if you look at the en face view, you can see it depleted here. I'll play this one more time so that you can see that in action. So this gave us the idea that maybe the actin is controlling the secretion. And so the only way that we could look at that secretion was by using a glass slide where we coated the stimulants for the T cell to undergo secretion, because that allows us to use a particularly rapid form of imaging that's fast enough to capture the granules as they're released. And so here are the granules that they're seen in red, and here's the actin in which you can see is depleted in the center as the granules are about to release, and the actin surrounding it. So what happens when the granules release? You'll see that there's a sudden burst as they release, but then virtually immediately the actin comes and covers it over, suggesting that the recovery of the actin is terminating the secretion. So to test this, Alex Ritter in the lab, when he was coming to the end of his PhD, used a drug called latrunculin, which can depolymerize actin. So normally actin is polymerized to make the cover around the edge of the cell. If you add latrunculin, it disperses that again. Excuse me, I just want to check. Hello? Sorry, I thought that was Ruth telling me she couldn't hear me. It's not. So what happens then is that when you add the latrunculin, the actin disperses and the granules then undergo a second wave of release. And as you can see here. So a new wave of degranulation occurs when you add the latrunculin, and you can see that here much more readily by the in the fixed frames. And you can then quantitate that, and you can see that if you just add a carrier, then nothing happens. You can cause this new wave by adding the latrunculin. So all of this meant that clearly actin is doing the job. So how could we study that in order to see what exactly was happening with the actin across the synapse? So if we look at T-cell receptor signaling and what's known about it, it's actually clear that one of the first events that happens when the T-cell receptor recognizes peptide MHC, be it cancer or any other, is it recruits kinases that activate an enzyme called phospholipase C gamma. And what phospholipase C gamma does is it cleaves one of the phosphoinositides in the membrane itself, called PIP2, to generate diacylglycerol. But that was of particular interest because PIP2 is known to be involved in recruiting actin to the outer cell membrane. So how could that then, maybe we thought that might be linking the T-cell receptor signaling to the changes in the membrane. So in order to study that, Chris Gordon Bone in the lab made a series of probes, or gathered them from other labs, such as Thomas Ballas, where you could tag something that would recognize particular phosphoinositides and with a fluorescent label, and you could then visualize all these events going on using the spinning disk microscope that I've shown you previously. So I told you that the first thing that happens is this phospholipase C gamma is activated, and so we could label phospholipase C gamma, and you can see that indeed, as soon as the two cells recognizes a cancer cell, then you get a clustering of phospholipase C gamma. That cleaves PIP2, and you can see that here is the PIP2 visualized with a different probe, and it depletes across the center, and it generates diacylglycerol, which you can see here. So that allowed us to see what was happening when you had actin there or when you didn't have actin there. And it was clearly that the membrane composition itself had changed because of the signaling events. So how could we work out the mechanism by which that happened? So by quantitating each of the different constituents and by measuring the timing of each of the different constituents. So when each thing appeared right after contact, so the phospholipase C gamma, the burst of diacylglycerol, the loss of the PIP2, and the loss of an enzyme that generates more PIP2, change in the charge on the membrane, and the loss of the actin itself all seem to be very, very tightly linked. So using this and a series of other approaches to test this model, we were able to come up with a model that really described very nicely how you can link the strength of the T-cell receptor with the level of actin depletion and the delivery of the granules. So here is what happens, that here is your T-cell membrane, and it has this, one of the components of all membranes is PIP2. When T-cell receptor activation occurs, phospholipase C comes in and it cleaves the PIP2 and makes diacylglycerol instead. Well, that changes the charge of the membrane, and the PIP5 kinase that keeps making more PIP2 to put back in the membrane can no longer bind because it binds by virtue of charge. So that falls off, and then that means that the actin has fallen off too because there's no PIP2 there, and that allows the granule to come through and be delivered. So you see that the whole thing can be very dynamically changed according to whether there's a little bit of T-cell receptor signaling going on. As soon as the phospholipase C gamma is gone, then it's all off again, and the PIP2 will come back, and the actin will come back. So this gives us some insights as to how some of the mechanisms that are absolutely fundamental to the killer cells work. But how can we identify further molecular mechanisms controlling the killing? So in order to do that, really, we needed to understand all of the different steps that gave rise to the killing and find new ways to identify completely new pathways that haven't been identified as yet. So some of the approaches that we've taken is we've used all the imaging to define a very high resolution map of the events that lead to killing, from the very first contact to the activation of different pathways to the delivery of the granules. And this is a very minimalistic map compared with what we have. And we also tried to develop screening techniques that would allow us to do more high throughput screens to look at differences in killing, either from immunodeficiencies, where we have lovely collaborations with Great Ormond Street and Newcastle to look at patients with immunodeficiencies where the CTL are not working properly. We use more standard laboratory techniques such as CRISPR knockouts or even inhibitors. But what I want to finish on is a study where we looked at single gene knockouts to try and identify novel pathways that were involved in killing, which in an unbiased fashion. So this was coordinated from King's College London and the Crick, and of course the Sanger Centre. So the Sanger Institute has been making a series of single gene knockout mice, and what a group of immunologists led by Adrian Hayday decided to do was to use their favourite functional models to test whether the genes knocked out in these mice affected any of these different pathways. And so twice a week during the length of this study, the different mice were sent in different directions in order to work out what the blood phenotype was, what the immune response in the skin was, what generation of anti-nuclear antibodies was. We undertook the cytotoxic T-cell screen, and then a series of infectious challenges or colitis model using DSS. And for us, this turned out to be very powerful, because it was as though if this mouse was a patient, it was sent simultaneously to many different clinics all at once, and all of the data was fed into a database that's publicly available. So for us, it was very rewarding, because from 500 single gene mutation mice, we identified genes that controlled completely different pathways that we hadn't looked at before. So some controlled apoptosis, transcriptional regulation, metabolism, protein recycling, the actin regulation, and there were some genes that are really orphan genes with virtually nothing known about them. But all of these appear to perturb the killing in one way or another. One of the unexpected results was that although we'd always looked for things that didn't kill properly, we actually found that by doing so many killing assays, we could identify gene mutations that led to enhanced killers or super killers, which is really rather cool, because identifying a gene that then leads to super killing is really exciting. So I just want to finish with one example where we identified a super killer. And in this particular case, you can see there's a very obvious phenotype in that the wild type have normal size secretory lysosomes, but these are gigantic in the super killer. So if you look by electron microscopy, here's the normal size of granule, and here's the super killer granule. So very simplistically put, it means that when they kill, each granule packs a bigger punch, and they do kill a lot better. So in summary then, what I've tried to show you is that combating cancer requires, of course, understanding molecular mechanisms and optimizing the recognition and signal strengths. Not the strongest is necessarily the best. And then therapeutics that can enhance pathways leading to killing. Now, I know that all of you will know that there's much more to immunotherapy than simply working out the best molecular mechanisms. And actually, one of the things that I've been involved in during the lockdown is chairing a forum of experts for the WISH Immunotherapy Forum, which will actually take place virtually in November. It's an organization which is non-profit, and it is sponsored by the Qatari Foundation and chaired by Lord Aradazi at Imperial College. And each year they put out, or each 2 years, they put out 10 different reports. And this year I've had the privilege of chairing the forum on realizing the promise of immunotherapy. And the aim of this report is to provide evidence-based examples for government funders and policymakers that will help to illustrate ways in which they can readily catalyze the discovery, the development, and the delivery of immunotherapies globally across many different economic backgrounds. So if you're at all interested, feel free to register. It's free, and I think it should be a very exciting session. So with that, I will just acknowledge the people in my lab who did all of the work, and point out, if you really like the movies, they're all available on our website. Thank you very much.
Thank you, Gillian. As last time I listened to you, absolutely spellbinding for me. And I just think immunotherapy is such an important part of the treatments we give, but this proper cell biology and drilling down on why is what's going to give us better immunotherapy as well, and maybe less toxic as well. Thinking about some of the CAR T toxicities. I've got a few questions there. One of them is actually my own, in terms of the strong versus weak signal. In your mind, is that what will explain sometimes with immunotherapy we get a stunning response, and sometimes it's slow and steady, and I don't know clinically which is better? You were inferring that weak and slow and steady might be better?
Well, yeah, so certainly from some of the mouse studies, that's what seems to be emerging. There are studies out there that support that, and that's what our studies would suggest. So I actually would love to know from the clinical side whether that seems to be true too.
Well, you certainly clinically see patients who respond quite quickly, and then there are some where they go into partial or complete response, but it can take a year of treatment with checkpoint inhibitors just to get there slowly in the tumor. So there presumably is. And Daniel Hochhauser sent me a question saying that do you think the phenomenon of different intensity stimulation producing a distinct downstream effect is specific to T cells, or could it relate to other immunological responses as well?
Oh, I think very much it could relate to it. Immune cells are, once they've found a good pattern, they tend to repeat it in different variations. So I would bet my bottom dollar it applies to other cells too.
And these centrosomes, can they do this in normal cells in terms of secretion, or is this a T cell thing? Because that's fascinating, just watching the movies of how they, are driving the events.
Yeah. So we love this discovery. So we discovered it, more than 10 years ago now, but it is used by other immune cells. It's not common. So actually what in most cells the centrosome does when it moves to the membrane is it forms a primary cilium. And so it's involved in celiogenesis. And so based on that actual morphological observation, we began to investigate whether there are links between how a T cell works and how ciliary signaling works. And in fact, Maika de la Roche, when she was in my lab, identified the hedgehog pathway as being involved in T cell activation signaling as well. So, and we found many, many more similarities. So it does happen in other cells, but to make a cilium. So I suppose I think of the immunological synapse as a frustrated cilium. So it's the immune cell cilium. But in both cases, you're getting secretion, you're getting signaling. So they're actually distinct biological systems which actually have a very similar mechanistic basis.
Well, thank you. And thank you again.